Welcome back, everybody. This is lesson 2 for week 2. Our topic is biotechnology. Before we start, we're going to get centered and ready to learn with our mindful minute. So, wherever you are, find a comfortable seat, sit with a mindful body, and we'll begin breathing for 1 minute together. Everybody, inhale through your nose, fill your lungs, nice deep breath. Exhale, pushing the air out. Inhale, nice deep breath in. And exhale, pushing the air out. Inhale, deep breath in. Exhale, pushing the air out. We're halfway there. Inhale, nice deep breath. And exhale, imagine pushing out tension and stress. Inhale, energizing oxygen. Exhale, stress and tension. Inhale, energizing oxygen. And exhale, stress and tension. Allow your breathing to return to normal. And check in with yourself, see how you feel. Notice if anything has changed. And when you're ready, you can slowly blink open your eyes. All right, welcome back, everyone. For our do now, you guys are going to name some of your favorite snacks. They might be pictured here, they might not. Go ahead and think, like, what's my favorite snack? Maybe it's Takis, maybe it's Cheez-Its, maybe it's sour gummies. All right, my favorite snack is probably popcorners, which are like those popcorn chip type things. I could eat a whole bag in probably 5 minutes. So, why are we asking you to pick out your favorite snack? If you've ever taken a close look at the nutrition facts or the labels on these snacks, you might see something that says this: partially produced with genetic engineering. What is genetic engineering? It sounds kind of crazy and maybe kind of scary, so we're going to discuss that today. Now, you might also see on some other types of snacks this non GMO. GMO stands for genetically modified organism. So, why are some things non GMO and why is that a big deal that it's labeled? So, our learning target for today is I can explain selective breeding and genetic engineering. Pause the video now so you can write down the learning target. All right, so we're going to watch a video on selective breeding, and along with the video is some check for understanding questions on your Google form.
In today's video, we're going to take a look at selective breeding, which is basically where you take the best plants or animals that you have in your population, and then you breed them together in the hope of getting even better offspring next time. Now, this idea isn't anything new. Humans have been using selective breeding for thousands of years, ever since we first started agriculture, which is just a fancy term for farming. Now, though, we use it all the time, and we selectively breed for all sorts of things. Like for cows that produce loads of meat or milk, for crops that are resistant to diseases, for friendly cats and dogs that make us lovely pets, and for plants that have large or unusual flowers. So, how does it actually work? Well, first, you take a look at the individuals in your existing stock. So, that might be all of the different tomato plants in your field. And you select the ones that have the characteristics you're after. For example, these two, with lots of big tomatoes. And then we can breed these ones together to give us the next generation, which will hopefully contain some even better plants. We don't just do this once though, we do it over and over again, always picking the best ones from each generation and breeding those ones together to get a better generation next time. To see just how much things can change, take a look at this picture, which shows the original form of sweet corn that we first found in the wild over 10,000 years ago, and compare it to this modern type. You can see this sort of change in all different plants, and it's just a result of repeated selective breeding, as we try to get better, bigger, and more juicy plants. As always, though, there are some drawbacks, and the main one is that selective breeding reduces the gene pool of the population. And if you haven't heard of the term gene pool before, it's basically the collection of all the different alleles held by an entire population.
All right, so he just used the word allele. Allele is a different version of a gene. So, for example, there is a specific gene that codes for the protein for our eye color. There are several types of alleles of that gene. We know this because there are blue eyes, brown eyes, hazel eyes, and different shades of color. So those different shades would represent different alleles of one type of gene, which would be eye color.
Whenever we do selective breeding, what we're actually doing is selecting for certain alleles that code for the traits that we want. So, as we do this over and over again, we're going to get a smaller and smaller pool of alleles as we get rid of all the bad ones and only keep the good ones. And because the best individuals are often closely related because they both have the good genes, breeding them together can sometimes lead to inbreeding, which makes the offspring particularly prone to diseases and inherited defects because their gene pool is so small. This is the reason that dogs like German Shepherds and Rottweilers are so prone to health conditions. A small gene pool also means that there's less variation within the population. So, if we were to go back to our tomato plants and imagine that a new pathogen arrived, which infected one of the plants, then chances are it could probably affect all of them. So, the farmer might lose his entire crop at once.
So this would happen because if that one pathogen came and infected one of the tomato plants and all the tomato plants were identical, then the chances are that pathogen would infect the whole crop.
And that's all for today. So, if you enjoyed it, then
Okay, so selective breeding. Let's look at an example before we get our definition. Over here, we have one type of cattle called the Brahman cattle, and it's bred with the English Shorthorn to give us the Santa Gertrudis cattle. So, what are the benefits of each of these cattle individually? The Brahman cattle has good resistance to heat, but poor beef. That says beef. Sorry, you can't really see it. And the English Shorthorn has good beef but poor resistance to heat, which means it can't really stand out in the sun for too long. So that could be a problem for a farmer. If now farmers figured out they can breed these two to get the desired trait, and the offspring would have the two desired traits. It would have good resistance to heat, so it could stay outside in the sun for a long time, and it would also produce good beef. So, selective breeding is choosing desired traits from members of the same species to breed better or more desirable offspring. You can pause the video now to write down the definition. Here's another example of selective breeding. Um, if you were to go to the grocery store and you saw green tomatoes, you are not likely to buy the green tomatoes. But they look big and juicy, but you're not used to eating green tomatoes. So scientists can breed these two types of tomatoes This one on the left for its red, desirable color, and the green tomato for its desirable size to get the perfect tomato with desired traits. The next type of biotechnology we're going to look at is called genetic engineering. Can you guess what genetic engineering involves? Let's find out.
Now, genetic engineering is one of those topics that can seem quite complicated at first, but it's really quite simple. The basic idea is that we find one organism that has a desirable characteristic, like a plant that has lots of big fruit. Then we take the gene that's responsible for that trait and transfer it to another organism, so that this other organism develops the same trait. So effectively, what we're doing is modifying the organism's genome by adding a new helpful gene.
So, a genome just refers to all of the genes inside an organism. So, they're saying they're modifying the genes in an organism.
And so we can call the organism genetically modified, or GM. We're not just limited to organisms of the same species, like we've shown here, though. We can also use the genes of one species to genetically modify organisms of another species.
So, genetic engineering is the process of changing DNA of living things for useful purposes. This is done by taking a gene from one organism and putting it into another organism for a desired trait. So, now that you've looked at both selective breeding and genetic engineering, what are some similarities and differences between the two? Take a moment and write this Venn diagram in your notes. And we will do one together. So, what are some similarities that we notice between the two? Well, I notice that these are both used for what? Getting desired traits. So I'll let you guys fill out the rest for the differences and similarities. Pause the video now and write this in your Venn diagram. So one of the examples in the video they talked about was creating insulin. If you recall from our body systems unit, insulin is a hormone that helps lower what? It helps lower blood sugar, and it's created by what organ in our body? It's created by the pancreas. So people with diabetes can't make their own insulin, and therefore they need to take insulin as an injection. So where do they get the insulin from? Well, it was actually genetic engineered and produced in a lab. So here's a human cell, and they take this gene that creates insulin and they cut it out. So oops, imagine that they took this gene and they cut it out. Then they take a bacteria cell and they take something called plasmid DNA from the bacteria. So it's one of the organelles in a bacteria. Don't worry, it's not like going to get people sick or anything. They're just taking this organelle and they are pasting the insulin gene into the plasmid from the bacteria. Then we have something called a recombinant plasmid, also called recombinant DNA, because they're combining two things: the gene that creates insulin and the plasmid DNA from bacteria. They take this recombinant plasmid and they put it back in the bacteria, and then they allow it to grow. Then the bacteria now has produced insulin that can be extracted and used for medicine. Now, why would scientists use bacteria for this process? Well, bacteria grows really, really fast. So, for example, if you have food out and it's not refrigerated, the bacteria can multiply in just about 20 minutes. So, bacteria grows super, super fast. That's why we always need to be washing our hands and being really careful not to spread germs. So, because it grows very fast, it is the perfect way to produce insulin in mass amounts to be used as medicine. And remember, using the bacteria doesn't mean that you're going to get sick or that it's dirty or germy because they take the insulin out of the bacteria and they extract it. So it's a safe process. So on the regions, um, questions involving genetic engineering will often look like this. So we have over here, we're going to analyze it together, the gene for human insulin, and it's put into, again, this cut bacterial DNA. And in the last picture, we know that this is called a plasmid. And it's cut and pasted into the plasmid by an enzyme. Then that cut and pasted plasmid is going to be put back into a bacterial cell, and then it goes through cell division because it'll multiply very fast and it'll create that desired protein at a very fast rate. So that's genetic engineering for you. It's basically like cutting and pasting things and then getting the desired trait that you want. All right, that's all for today. For your assignment, you are going to, one, fill out these questions that go along with the lesson, and then you're going to work on part 4, which is an assignment that is all regions questions related to selective breeding and genetic engineering. See you guys next time.